So the purpose of this webinar really is to talk about the access-related issues of BBMRI-LPC. This concerns really, I guess the real coordinated meat of what this this whole program is about, and it concerns about half of the work packages. And I'll be going through each of those a little bit in more detail. So just one quick thing, I'm going to go through this webinar, which will give the background about BBMRI-LPC as well as discuss the access issues related to the project. We're asking everybody who has questions to send them in to us while I'm talking, and we'll try to coordinate those questions at the end. And people will also be able to give answers as well. So if you want to talk, either to ask a question, to make a point, or to give an answer, you have to write to us, and we will then, we can give each individual the opportunity to speak. We have to turn you on, put you in speaking mode from here in Lyon. And so hopefully this will work fine. So just many of you are very aware of BBMRI-LPC. Some of you are not so aware. So the first thing I'm going to talk about is just give you a quick background to BBMRI-LPC. The aim is very much to bring together large prospective cohorts in Europe and basically use them for groundbreaking science and facilitate transnational research projects across Europe. This came out of a call in 2012. There were two separate projects, and at the strong suggestion of the European Commission, we brought these two projects together. Basically, just a few obvious things. Why collaborative prospective studies? Why should we bring them together? Well, for many of us, it's obvious that prospective studies are absolutely crucial when trying to establish the underlying causes of disease. Risk factor information is obtained before disease onset occurs. It helps avoid the issue of reverse causality. We need to bring prospective studies together often to have large enough sample sizes to provide validation of initial results that might have come from smaller studies. And also, we often need to bring studies together to study rare endpoints. I just have a couple of examples that I'm going to go through quite quickly. This is one of the first large prospective cohort studies initiated in the UK by Doll and Hill in the early 1950s. And it's proved fundamental in thinking about the way in which exposures like tobacco have such a strong influence on health, and it's also proved fundamental in introducing restrictions on things like tobacco as well. So it's a good example of a large, well, what was considered for the time anyway to be a large epidemiological study that's had almost a revolutionary effect on the health of the population. To just give another example here, this is an example of the effect of hypertension on mortality, and it's an example that was given to me by Sir Rory Collins in the UK. And so it's looking at the relationship for different levels of systolic blood pressure by age on the risk of coronary heart disease. And I think these results are simulated, but based on a cohort of 5,000 people, really it's very hard to see any trends there based on the small numbers of outcomes with increase in systolic blood pressure and risk of coronary heart disease. If you look at a cohort of 50,000 people, you start to see some trend. The numbers are still quite all over the place, however, and it's only when you look at 500,000 people that you see for each age group, you see dramatic increases in risk with no threshold effects. And this was always the question, whether there is a threshold effect with systolic blood pressure and the risk of coronary heart disease. And obviously, I think that these results were very important in showing that there really doesn't appear to be any apparent threshold effect at the lower levels of systolic blood pressure. Just another example, this is an example from our field where we're working on a rare outcome, oropharyngeal cancer. Within the EPIC cohort, there's about 140 oropharynx cases that have occurred among the 500,000 individuals within the EPIC cohort, and based on this, we've been able to evaluate the strong effect of HPV serology on subsequent risk of oropharyngeal cancer. This is also a good example of a study which, even though it's based on 500,000 people, the numbers are still quite limited. And to investigate this further, one often might need to bring together even more large cohort studies. This is another example of genetic association studies that compares the results for lipids based on studies of 2,000 versus 100,000 individuals. And when one considers 100,000, well, obviously when one considers just 2,000 individuals, one does see suggestions for strong findings, but most of these are just suggestions, and there's only one or two findings that would be considered significant at the traditional statistical, using traditional statistical criteria. But when these results are pooled with many other studies to reach a far larger sample size, then the results are obviously just so much more robust and so much more convincing. So obviously with these genetic type studies, it really is this idea of bringing together many studies just to have far larger numbers. There are, I think it's worth mentioning, there are other initiatives that bring together cohorts. There is the NCI cohort consortium that some of you will have been involved in. It's very different to what we're trying to do here, but I think it's worth mentioning. It's basically an umbrella organization which is coordinated from the US NCI, and it gives the possibility for about 50 different cohorts around the world to discuss, plan joint studies, etc. And in some ways it's been very successful. Some examples are the Breast and Prostate Cancer Cohort Consortium, which was mainly established to investigate gene-environment interactions. If we just look at the output that's published, there's been 42 peer-reviewed publications, some of them extremely high-profile publications that have come out from that cohort consortium. There's also a Pancreatic Cancer Cohort Consortium. This is another area where on its own it's difficult for any individual study to really produce robust results on pancreatic cancer, given that although it's a very deadly disease, it's quite a rare cancer. So many different cohorts have come together to investigate genetic and lifestyle risk factors. And again, that's another example of a successful consortium. Here's just a few other examples of consortia that are underway. There's one that we're organizing here from Lyon that involves 21 cohorts. It's basically involved in evaluating the role of B vitamins and lung cancer risk, and involves around 12,000 samples. And there's a head and neck cancer consortium underway as well. So I think for those of us that have been involved in establishing these consortia or working with colleagues, even though we can see that the collaboration, once it's up and running, can be incredibly fruitful, and it's something that is really of immense benefit to all who participate, there are problems in establishing these consortia. Problems include issues like access costs. The cost of retrieval of blood samples from consortia is often very high, and this is mainly just due to the fact that studies need to charge for this type of work. Biorepositories tend to be to involve a lot of personnel. It involves a lot of time. Often one needs to support investigators in each cohort. There are issues such as database harmonization as well, because databases tend to be very different between cohorts, and to conduct a common study together, a certain level of database harmonization is required, and this is often done separately for each individual collaboration. There are administrative obstacles as well for when bringing cohorts together. One will need to obtain ethical approval from each cohort, and this might require different things. There will be material and data transfer agreements that will be different for each cohort. And if we look at the example of the NCI cohort consortium, the NCI basically just provides the opportunity for cohorts to collaborate. It does not, however, provide any additional support. It's also a consortium that's very much, as one would expect, focused primarily on cancer, and so I think the idea is if we are to initiate something that's bringing cohorts together in Europe, is there a way we can improve on what's currently available? So the overarching goal then of BBMRI-LPC is really to pave the way for future large-scale projects in Europe that can be initiated by European investigators. So to help clarify legal and practical obstacles for conducting pan-European prospective studies, and also just demonstrate that it can be done efficiently. And that's efficiency with regards to the time, the effort it takes, as well as the financial resources that it takes as well. The focus is very much on science, so hopefully this will help support really high-level research projects across the prospective cohorts. And also, I think by bringing together investigators from different cohorts, it will create a forum for large European prospective cohorts where we can increase communication, provide a forum for discussion of common infrastructure priorities and scientific opportunities. Just regarding some of the specific aims of BBMRI-LPC, and today we're only going to talk about the first four of these, but the specific aims are included here. So improving harmonization of individual data on health, lifestyle, etc. Harmonized definitions of diseases, so we have specific work packages for each of these. Provide free transnational access by users through study proposals selected by an open pan-European call. And in the framework of these studies, generate and provide access to whole genome sequences and other omics data. So these four top specific aims are really what we're going to be talking about today. And the other aims, they are part of BBMRI-LPC, but they're not topics that we're going to discuss in detail today. Okay, so we're going to discuss the four specific work packages that are covered by these top four specific aims. And this basically, these work packages basically form the whole access component of the project. And they're outlined, and how these different work packages work together are outlined here. So we have work package 5, which is involved with exposure data harmonization, coordinated by Nadia Slimani here in IARC. Work package 6, which is harmonization of clinical endpoints, which is coordinated from Imperial, Elio Ribili. We have work package 4, which is the actual access to samples, facilitating access to samples and data, and the financial reimbursement for this, which is coordinated by Eric Wickman in Munich. Work package 8, which is the access to what we call here upgraded data, which is coordinated from Barcelona by Ivo Gut. And we have work package 10, which is the work package I'm coordinating, which is basically on the establishing the cohort research infrastructure network. And we're going to talk more about this later. So we'll start off then. I guess when the project was put together, we didn't think too much about the having these work packages in the right order. So we'll start off with the last one, which is the work package 10. It's called the Cohort Research Infrastructure Network, and it's basically responsible for coordination of these various access activities. And this is done through a cohort coordinating group that involves PIs from each of the work packages involved in access. And also, I think, not to make it too cumbersome, we did want to have strong involvement from the participating cohorts, but we have stipulated that this should be 4 PIs from individual cohorts. So this will be the cohort coordinating group that will soon be up and running. And the aim is to enable the scientific projects to be proposed, to be approved, and be put to the individual cohorts. So we will actually involve peer-reviewed scientific calls for proposals that will be open to any scientist within Europe, and there'll be more details on this later. So we will prioritize and evaluate these scientific proposals, and we'll hopefully facilitate the management of access and identify any particular barriers. So we'll be going through some more of these issues in a moment. Work package 4 is regarding how one facilitates the access to the samples and the data, and this is specifically from a financial point of view as well. So there is a lot of support for the researchers to actually have access to the data and have access to the samples, and we'll talk a little bit more about how this is calculated later on. But basically, once a project is approved, then it's put to the individual cohorts for the participation of the individual cohorts. Obviously, if a project is approved by us, we'd hope that as many cohorts as possible choose to participate in this, and the idea is that the funds you receive go towards these approved projects. And then it will be up to individual cohorts to provide the access to the samples and the data, etc. Okay. The work package 5 and work package 6 will run almost in parallel, and they're basically to support harmonization of exposure data and clinical data, and this is done as part of the ongoing projects. So it's basically, we envisage here, I mean, obviously there's an awful amount of work that could be done on data harmonization and harmonization of clinical endpoints, etc. And one will need to be selective in exactly what exposure data is harmonized, what clinical data is harmonized, etc. There are some interesting questions that we have with what we should do with the data that is harmonized, where it should be stored, how it should be accessed, etc. Should data be kept at each study center, or should it be kept at one specific center, and we somehow allow access for investigators from a remote site to access this data on a server? Or should we send data to individuals once their project has been approved, etc.? And so at the moment we haven't decided on any of these issues. We do envisage some a research infrastructure that will involve a web-based micro-opal situation, and this is provided by the Maelstrom Research Group in Canada. Okay, so this is something else that we will be developing. Just talking about some of the initial requirements for Work Package 5 and Work Package 6, I think you will be requested to provide by the PIs of each of these work packages data inventories in English, providing a list of various domains, etc., and obviously to identify the contact within each cohort who can be contacted regarding the type of data in each cohort. Similarly, for Work Package 6, for the clinical endpoint data, each cohort will be requested to provide clinical endpoints and identify the contact who one can contact to provide extra information on clinical outcomes. So these are steps that are going to start in the next weeks, I imagine. With regards to Work Package 8, the access to upgraded data, this basically aims at providing additional omics type data as part of the overall BBMRI-LPC program. How we actually define this is still to be decided. What should, it can be done in conjunction with our initial scientific calls. It can be done as part of an overall strategy to, on a small part of the population, provide a very omics-rich data on this subpopulation. It's something for the moment that we have not decided, but there is a budget there, And I think at some point we have to decide how this is going to be done. Just one or two things about the scientific calls. There is the cohort coordinating group. As I mentioned before, is made up of the 5 work package PIs, as well as up to 4 cohort PIs. So that makes a membership of about 9, which we think is still more or less manageable. And the work of this group will be to identify priorities for the research calls. So we have the possibility to say that we would request research calls on specific areas. We will establish criteria for the sort of calls we're looking for, and we think that any calls that come in should basically benefit from the multinational, large prospective nature of this collaboration. So calls that could be done on small retrospective studies, or projects that could be done on small retrospective studies, would obviously have less priority. An important aspect is that any funding that is available within this project is for access. It's not to actually fund the research, and it's quite clear we don't have any funds to fund the research. So research projects will have to be funded independently. So the cohort coordinating group will carry out an initial evaluation of research projects to establish some kind of priority, and then some pre-selected proposals will then be submitted to each of the individual cohorts for your individual review and hopefully your approval. I mean, obviously this is just so no cohort will be obliged to participate in any project for whatever reason they see fit. They can decide not to. So this whole initiative is really just to facilitate the participation of as many cohorts as possible in projects that we see as being of high potential and high priority. But in that, there is in no way any obligation for any cohorts to participate should they decide not to. We will generate standard operating procedures for how to access data and how to access samples. These are currently being prepared. We'll also provide standardized descriptions of the available cohorts and what samples and data are available. This will be an extension of the current BBMRI catalog, and we will advertise the calls, and we plan to make our first call next spring. So that's the background to the access. Obviously, the participation of as many cohorts as possible is crucial. We need to know what cohorts are available, what the sample size is, what the number of and type of biosamples available is, what the follow-up time is for each cohort, what sort of endpoints, etc. We also need your participation really to estimate how much it costs to access data and to access biological samples, and what your own internal review procedures are. So I think it's important that we can make a lot of this information as transparent as possible to help people decide what projects they want to propose. And we also want you to consider the research proposals that we pre-select after the scientific calls are made. There are some financial resources for the cohorts, and these are really just in order to enable you to provide access to the data and to the biological samples. So funds have been transferred or will be transferred to cohorts based on the estimated costs of providing access. There's about 1.1 million euros in total that's available for this activity, and this really has to be the real access costs that are calculated by each individual cohort. I mean, it's an important point here that when cohorts receive funds, if these funds are not then used to, or if not all of the funds are used to provide data or provide samples, then the European Commission will request that these funds or any unused funds are reimbursed. So it's quite a complicated procedure for these access funds. I think it's something that, according to the European Commission rules, it's something that they wanted to define it like this. But basically, we will be forwarding funds to each cohort based on the estimated required amount, or how much it costs to provide this access, and any unused funds will have to be reimbursed. So this gives an idea of the amount of funds available for providing access to the individual cohorts. These are estimates. They're based on the initial estimates of what the costs were for providing a certain amount of biological samples or a certain number of data units to the approved projects. And as I said, if these funds if they're not used, then we will not ask them back, but the European Commission will. Okay, so that's the other cohorts that are participating. Obviously, this will involve some work from participating cohorts. We're requesting that each cohort provides information. That this obviously takes time. We're requesting that you participate in the review of proposals. There's not necessarily any immediate advantage in terms of funding, apart from obviously funds that will cover the access costs. But we do hope that really that all cohorts will participate in this, and we do think very much that it will be of benefit in the future to all participating cohorts. Obviously, any cohort that's participating in this can also propose their own topic to be considered by the cohort coordinating group, and then all of the other cohorts. Just one clarification on this point. Access costs can only be used to provide access to samples not in the country from where the application originates. But we do think that there will be a long-term gain for participation in this initiative. I mean, it's clear that many cohorts at the local level, at the national level, are facing funding difficulties. We recognize that there's a need for long-term infrastructure support. There's needs for cohort development. I think this message is starting to get through. I think more and more groups like the European Commission, for example, are realizing the importance of developing cohorts. I think that this was funded is recognition of this. It's obviously difficult for any single cohort, perhaps impossible for any single cohort to attract major support on the European level. And this whole initiative, we hope, will give us the opportunity to develop an EU-wide strategy, not only for the science of what happens within cohorts, but also for funding the network of prospective cohorts. That's the long-term strategy. There are obviously strong scientific motives for bringing the cohorts together. Some questions can only be studied within cohorts, and hopefully that this will ease a lot of the obstacles that exist in bringing together multiple cohorts, and it will prove to be cost-effective and will lessen the administrative burden and logistics. This is my final slide. Within future perspectives, we hope that there will be future perspectives within Horizon 2020 that will allow us to further develop this initiative. There is obviously the strong international focus on non-communicable diseases, non-communicable and chronic diseases, and which is obviously especially important for an aging European population. I think this is only going to become more important. We think that this collaboration could be an excellent opportunity to put forward a way to really study the health of the European population, especially where it refers to chronic disease and common lifestyle risk factors. And one issue that we've already started talking about is whether, based on our current work, whether we can move forward and at some point develop a more formal European cohort consortium. So those are the slides that I wanted to present. Now I think, hopefully some of you have been sending in questions. I think we've received quite a few, and one by one we're going to try and go through these and give the possibility to talk over to different investigators. Okay. So first will be, well, I'll let Matthias say, because he's been looking at the different responses that have been coming in. Perhaps, I don't know if Marcus is online or Gert-Jan, if they wanted just to say to say anything. Okay.
All right. So we're going to give Gert-Jan van Ommen, one of the coordinators of the BBMRI-LPC project, the word now right after. So Gert-Jan, we're activating you now.
Good. Am I?
Yeah.
Can you hear me? Okay, good. One thing that I wanted to clarify to make this business of the access a bit clearer is that the access that has to be provided needs, as you said, to be transnational. So that means you have to have people that propose from one country and people that provide the cohorts from another country. Now, typically when you use more cohorts, this is a condition which is easily to be fulfilled. But the other thing that at least was made clear to us, and is also similar to one of the other EU projects that has this access component, is that the key is to provide people from outside the primary participants this access. So that means that this is not about research that different cohorts would want to do with each other. So if there are different cohorts within this BBMRI-LPC, it won't work, or at least it's reasonably frowned on when sort of 2 or 3 cohorts would together write a proposal and say we want to study each other's samples.
Okay.
Anyone there?
Yep.
So the fundamentally what this should be about is that there is nothing against proposing ideas to people that you know, and there's nothing against being approached by people from different cohorts, but ideally this type of funding is for providing access of the European scientists to the cohort material. The benefit for the individual or joint cohorts would be that they actually in this process get their samples enriched with additional clinical or omics or epidemiological data.
Okay, thanks, Gert-Jan. I've been thinking about this point as well, and sometimes there is an issue that the people who work on cohorts tend to be the people who publish results on cohorts as well, and I think we're just going to have to wait and see what happens when we release our scientific call, if we will receive, good, strong applications to have access to the data and the samples from people who are not currently working on cohort studies. Obviously, that's the aim, but whether this is something that will happen to a large extent is something we'll just have to wait and see. Okay, so I'm just looking. So if you do have questions, then please write them in. We have a few questions that have come in from Sweden. Hold on. Okay, no. So we have a few questions that have come in from Sweden from from Nancy Peterson. One comment was more comments really, is that things like cohorts and outcome information is a moving target, and how will cataloging activities be updated. Obviously, if this is a long-term effort, we will hope that these sorts of activities are updated. It's again, we need to make this a long-term effort. Do we have any other questions coming in?
So whether they can get support for linking data with registries. So this is a good question.
Yeah, I guess this would be, I'm not quite sure of the answer to this question, because it's another question from Nancy about whether cohorts can use some of their access funds to link their cohorts to local registries to obtain clinical information. It's a good question. I don't actually have the response to that at the moment. I think I've just received a message from Marcus that we'll have to ask this from the Commission. So we'll seek clarification from this point. We've also received a question from Michaela Mayhofer, who asks, what happens to data after BBMRI-LPC? Again, I mean, it's a good question. I'm not sure if this is the sort of information that, or the sort of criteria that we stipulate from BBMRI-LPC. I imagine that all cohorts have their own criteria for what happens to their data after they've given permission for it to be used and it's been used for that purpose. Normally, one would expect that the data set is no longer used. I mean, the data will obviously be returned to the cohort, but that's something that we can discuss more.
I think any use of data in any specific research project would always be sort of governed by a material transfer agreement, and the rules would be set in a similar way that you would do in normal cases. So when it comes to samples, for instance, you would expect them to return or to be destroyed and so forth, and similar rules for when it comes to data only.
Okay. Do we have any other questions? Okay. I don't see at the moment any more questions coming through. Okay. Oh, we'll just give the so if there's no more questions, Marcus has asked for the opportunity to say a few words. So Marcus Parola from Helsinki. Okay, Marcus.
Hello. Yes. Hello, everyone. Thanks for joining the webinar. I think we will have to do more of these in the future when we're closer to the calls. Anyway, thanks for Paul. First of all, nice presentation, and a couple of things. First, related to your presentation, and then to the issues that have risen up. In my view and in my mind, BBMRI-LPC is kind of a spin-off of BBMRI-ERIC, and we are doing in LPC many of the things that ERIC will take on after the LPC. So we're kind of partly preparing for BBMRI-ERIC to be fully in effect, while of course in the same time we are an independent project that has its own responsibilities and ambitions. But that was kind of said in Paul's presentation, but I just wanted to make it a bit more clear. So if you follow American media, so if Cheers, BBMRI-ERIC is Cheers, so we are Frasier, in a sense. Probably parts of LPC will integrate in ERIC in the future, but let's see what those are. Then really hot news or fresh news from Work Package 8. I just met at Boston Work Package 8 co-leader Arno Palotia, who's leaving from Sanger to Boston Broad Institute, and but Sanger will still remain as the work package 8 leader. And I had a meeting with Richard Derwin, who was very interested in contributing to this. And as most of you probably know, Richard is one of the, let's say, he's the doctor sequencer of Europe in a sense. So he's really like in the forefront of sequencing business. However, his suggestion last Friday actually was that given the limited resources, that is money we have for sequencing now, we couldn't do much with sequencing, much novel science in a sense, because there are much more large projects already going on. And what he suggested that we should actually go for genome-wide genotyping, especially from cohorts that don't have such data as of yet. This is just came up, and this really hasn't been discussed in the steering group or anything. I just wanted to let the cohorts know at this point that this is one of the potential happenings here. So the idea would be that we would do only few samples per cohort deep sequencing, and then do one of these new biobanking chips that both Illumina and Affymetrix are working on. And given the price, given the number, we could get a really good price on those, and we could actually genotype significant numbers of those cohorts that don't have such data as of yet, and such build to the European infrastructure of biobanks some important data. So, but that's still in the level of idea, and nothing has yet been decided on. So, okay, so I guess that those were my two points I wanted to make, and again, thanks for participating.
Okay. Thank you very much, Marcus. So I think if anybody has any additional questions, then please continue to send them in, and we'll answer them, or we'll get back to you individually. We've just had one last question. Maybe I'll just handle this one before we finish off. It's a question from Marcel Goldberg. What happens when access rules to BBMRI-LPC are in contradiction with specific rules of an individual cohort? I think one of the points I tried to make clear was that a cohort is never obliged to participate in any particular project, and these are just projects that are put forward to the individual cohorts after a certain recommendation, if you like. And obviously the individual rules of a cohort will trump any suggestion from BBMRI-LPC, LCP. So I think we can finish off there. So thanks everybody for joining the webinar. I hope you found it useful. If you have additional questions, then you can continue to send them in, or you can send them to us directly, and we'll try to deal with those as they come in. So thanks again everybody for participating, and of course we'll all be in contact again soon. Okay, so we'll end the webinar there.